Welcome children. This is another video for class 12th biology subject, and the topic we are going to study in this video is 6.5.1 transcription unit. If you remember in the last topic, in the last class, we studied that transcription involves copying of genetic information from one strand of the DNA to form RNA molecule. So in transcription, only a segment of DNA is copied to form the RNA. Not only that, another difference is that only one of the strand of the DNA is copied into the RNA. So only a particular segment of the DNA is being transcribed, and that too is transcribed only from one strand of the DNA. Therefore, it is very important to define that particular segment, number one. And number two is to know about that strand of DNA which would be transcribed. So that is what we are going to talk about. The first necessity, that is what is meant by transcription unit. So if you define transcription unit, a transcription unit is defined as a particular segment of DNA molecule which will be transcribed to form RNA molecule. In a transcription unit, there are three regions. In a transcription unit, there are three important regions. What are those three regions? There is a promoter, there is a structural gene, and there is a terminator. Now, please focus on this term, structural gene. Now, you should always remember that structural gene is one of the part of transcription unit. What is transcription unit? It is that complete segment of DNA which will be converted into the RNA molecule at the time of transcription. So structural gene is one component of the transcription unit. These are the three regions of transcription unit. Now we are going to study that the two strands of DNA, out of two strands of DNA, only one DNA strand gets transcribed. So what are the rules? What are the rules which define the two strands of DNA of the transcription unit? That means which strand of the DNA will be copied into RNA and which strand will not be copied into the RNA. Now, before that, the enzyme which is used for transcription is known as DNA dependent RNA polymerase. The question is why it is so called? Why it is called DNA dependent? Because it uses one strand of DNA molecule as a template. Hence it is called DNA dependent enzyme. Now why it is called RNA polymerase? This is called RNA polymerase because it polymerizes the ribonucleotides and forms a RNA strand. So there is polymerization of the ribonucleotides to form a single-stranded RNA molecule by using one strand of DNA as a template. Hence it is so called. What is the property of this DNA-dependent RNA polymerase enzyme during transcription? It can polymerize the RNA strand only in one direction. If you remember the property of DNA-dependent DNA polymerase during DNA replication, the same property was there. That enzyme was also replicating the DNA strand only in one direction, and that direction was 5' to 3'. So the same property is found in the DNA-dependent RNA polymerase also. So let us now define the two strands. The strand which has the polarity 3' to 5', the DNA strand which has the polarity 3' to 5' will act as a template. That strand of DNA will act as template and hence is referred as template strand. So remember that which strand of DNA molecule will be regarded as template strand? That strand which will be having the polarity of 3' to 5'. Hence, from this DNA strand, new RNA strand will be transcribed. whereas the other strand of DNA which has the polarity 5' to 3', and remember this formula, that the other strand of DNA will have the sequence same as RNA that will be transcribed. Now that other strand is actually displaced during the process of transcription. Ironically, this strand of DNA, which actually does not code for anything, is referred as coding strand. So here we have therefore defined the two strands of DNA. One strand is known as template strand and the other strand is known as coding strand. Out of these two strands, which DNA strand makes the RNA molecule? The strand is template strand. And this template strand has the polarity 3' to 5', therefore new RNA will be synthesized in 5' to 3'. Now this is another important point, that how do you know that which strand will be regarded as coding and which strand will be regarded as template strand? So all the reference point, while you are defining a transcription unit, all the reference points are made with respect to coding strand. Now to explain this point, a hypothetical sequence from a transcription unit is explained below. You have been given this double-stranded DNA molecule. As you can see, this strand has polarity 3' to 5', so it is known as template strand. The other strand has polarity 5' to 3', so it is referred as coding strand. So the question is, can you now write the sequence of RNA transcribed from the above DNA? We can write the RNA transcribed by knowing the formula that template strand is the strand from which the RNA is being synthesized in the direction 5' to 3'. So this is the RNA molecule which is being transcribed. I have written it for your convenience at the bottom. Now you can see that from this DNA strand, which is called template strand, this RNA molecule has been transcribed. And you can see the RNA molecule which is being transcribed has the same sequence as that of the coding strand, except since this is the RNA, so it will not have thymine. Instead of thymine in the DNA molecule, the RNA will have uracil. Otherwise, the sequence of coding strand is exactly similar to that of the RNA transcribed. So this is how we have made the RNA molecule. So similar questions you may be asked in board exam. So what you have to keep in mind, that you are making an RNA molecule. So keep the track of writing uracil while pairing it with the adenine in the DNA, and the polarity has to be written because RNA is being transcribed in the direction of 5' to 3'. So the polarity has to be written and the basis should be clearly written. Now we were talking about the transcription unit. This is the figure which is telling you the different labels in a transcription unit. So the first region is known as promoter region. This is shown in the box, red box. On the other end of the transcription unit, you have another region known as terminator region. This is in the figure shown by the purple box, and in between the complete region is known as the structural gene. So what are the three regions? Number 1, promoter, number 2, terminator, and number 3, structural gene. These are the three regions of a transcription unit. And we also studied that the strand having polarity 5' to 3', the strand having polarity 5' to 3' is referred as template strand, and the strand having polarity 5' to 3', sorry, the template strand having polarity 3' to 5' is referred as the template strand, and the DNA strand having opposite polarity will be referred as coding strand. So from which strand the DNA will be used for making the RNA? This strand will be used to make the RNA molecule, whereas this strand will not be used for transcription. But the reference point while defining the different things in the transcription unit will be made with the coding strand. Now let us see what is the location of promoter and terminator. In general, promoter and terminator, they are present on the opposite sides of the structural gene in a transcription unit. So first of all, let us see the location of promoter. Promoter, as you can see in the figure, is located towards the 5' end, which is referred as upstream of the structural gene. Not only the structural gene, but exactly it is upstream of the coding strand. So coding strand, its 5' end is this, and therefore this direction of coding strand is referred as upstream. So where is promoter located in the transcription unit? Promoter is located towards the 5' end of the coding strand, which is referred as upstream. And what about the terminator? What is the location of terminator? Terminator similarly is located at the 3' end of the coding strand, which is known as downstream of the coding strand. Okay. What are the functions of promoter region? It is the DNA sequence that, number one, provides binding site for the RNA polymerase enzyme. The same RNA polymerase enzyme which is involved in the process of transcription. So the first event is that the enzyme RNA polymerase should bind with the promoter. Number two, it is the presence of a promoter in a transcription unit that also defines the template and coding strands. What does this line mean? This line means that if the enzyme does not bind with the promoter, instead of binding with the promoter, if the enzyme binds in the initial stage with the terminator, the definition of strands will be reversed. This is what is written: by switching its position with the terminator, if the enzyme instead of binding with promoter binds with the terminator, what will happen? The definition of coding and template strands will be reversed. This is the significance of promoter, and this is the location of promoter in the transcription unit. What about the terminator? I told you it is located towards the 3' end, that is downstream of the coding strand, and the function of this is that it defines the end of the process of transcription. So as the name suggests, terminator, it terminates the process of transcription process.